decided that we would have the exam next Friday, correct? All right. So it starts with the gymnosperms, and I'm going to double check the schedule. But initially, I was thinking to have it go through this slide set on Phloem translocation. But let me double check that. I don't like to have the exams too different in terms of point value. So I got to make sure that if we stop at Phloem, I don't want the final to be overwhelmingly big. So let me look at the schedule a little bit more. But when I was first looking at it, I thought, well, maybe we could stop after Phloem translocation for a second. But I think we left off last week, we were looking at this idea of, the sugar movement through the plant. And it's, even though it's right next door to the xylem, the sieve tube elements are all clustered there next to the xylem. We have in the xylem transpiration, we have water being sucked up the plant from the leaf, generating that force and doing a tension gradient. Whereas right next door in the sieve tube element, we have a positive pressure gradient going on, and it can be pretty much in any direction through the plant body. Whereas with the xylem, transpiration is just one way, in through the roots, up through the stem, out through the leaves. And it just goes up, up, up. Phloem translocation, it's pretty much wherever the sugar is needed. And so we say that it moves from source tissue to sink tissue, source to sink. So we looked at this idea of it's really based on water potential concepts. So we know that water potential moves, water will move from water potential areas that are less negative to more negative water potentials. Okay, and so now with the sieve tube element, once you have sugar that's dumped into the sieve tube element, it's a little different story, because when we start talking about water potential movement, we're often thinking about osmosis, which osmosis is a two-part definition. It's the movement of water across a membrane, a selectively permeable membrane. Once you have sugars dumped into the sieve tube element, I just want to caution you or remind you that in these regions where the sieve tube elements stack end to end and join up, but those are the sieve plate ends of the sieve tube element, those are not, there's no membranes there that crossed over, so that doesn't happen. You don't have a membrane that water has to cross through, or the sugar solution in this case. All right, so once you start thinking about what's happening in the sieve tube element, it's really, we're looking at a pressure gradient that establishes from source to sink. And so here, let's go, I'm going to break this down. So I like to think of it in two phases. Phase one is thinking about the source tissue, which is usually the leaves that are doing lots of photosynthesis. And then once they make their sucrose, they have to get that into the vein if other parts of the body need it. So that's what you're seeing here. The red balls are the sucrose, and they're getting placed into the sieve tube element. And the companion cell, remember, there's a one-to-one relationship between companion cells and sieve tube element partner. So you see the sucrose getting loaded in. Well, anytime sucrose is a solute, anytime you dissolve solutes in water, that drives your psi sub S to be more negative, okay? So more solutes, that S stands for solutes. So more solutes, more negative, okay? More solutes, more negative, more solutes, more negative. So if you, whoops, that's supposed to be, oh, I can't spell and talk at the same time, clearly. More solutes, more negatives. Sorry for the sloppy writing. More solutes, more negative. Okay. So you see your psi sub S value has dropped to negative 1.7 megapascal. Well, that's going to make your overall water potential more negative, because water potential, especially when you start thinking about what's going on in the plant body, we tend to zoom in on what's happening to the psi sub S and what's happening to the psi sub P. Add those together, and that gets you your overall water potential. Okay, so if you get your size of S more negative, that's going to also cause your size of W to become more negative. Okay, then what happens very quickly after that is water moves from less negative water potential to more negative. So when you have this more negative water potential in the sieve tube element compared to right next door in the xylem, you're going to have water movement from less negative to more negative. You look at the size of W, or they just label it as psi, should be size of W. It's negative 0.8 in the xylem right next door, and then the Phloem sieve tube element is negative 1.1. So less negative to more. Okay. So then water rushes in right after the sugar gets loaded in, and then as a consequence of that, your size of P value starts to go up. All right, so it's just like this domino, boom, 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 this process where you have the sugar loaded in, that drives your size sub S more negative, that causes your overall water potential to be more negative, that causes water to rush in from less negative to more negative, rushes into the sieve tube element, boom, there goes your size of P, nice positive size of P. So now you're establishing a positive pressure in the source. So I wrote that down here. Increases the solute concentration in the sieve tube element. That makes your size of S and size of W to become more negative, and then you get water uptake. The water will flow from the less negative xylem water potential to the more negative sieve tube element water potential. So now that you've completed the story, that okay, your size of P, you've established a nice positive size of P in the sieve tube element at the source. Have I convinced you? Does that kind of make sense? But that's not the end of the story. So all that we've done here is we've described how positive pressure is being generated here. But now we need to think about a gradient, because that's how you get things to move. It has to be a gradient that it follows. In this case, a higher pressure to a lower pressure. So somewhere we have to describe a lower pressure situation compared to this source. So now we look at phase 2 with what's happening in the sink. So maybe it's that potato tuber that's growing at the end of your garden season, where the tubers are starting to pack away sucrose into starch. And so that's what you're seeing here. So that green cell is your sink cell. Think of that as your potato tuber. And you see that sugar is getting out of the sieve tube element into the actual tuber. Okay, well, now it's kind of the opposite of what we just described in the source. If you have a bunch of sucrose leaving or exiting out of the sieve tube element, that means your size of S is going to get less negative, less solutes, less negative size of S, less solutes, less negative size of S. Okay, well then that makes your overall water potential less negative. So now you can see water's moving from the less negative sieve tube element to the more negative xylem right next door. So based on the water potential difference there, you see in the sieve tube element, it's negative 0.4. Over in the xylem, it's negative 0.6. So it's less negative to more negative. Well, if you have water being exiting out of the sieve tube element, then your size of P is going to start to drop. Less hydrostatic pressure. Okay, so now you've completed the story. You're showing that in the sink, at the tuber, there's less pressure. At the source, there's more. Okay, so until you complete phase 2, now you can start saying, okay, yeah, there's a pressure gradient. And that is how the sucrose solution will move. It's going to move from high pressure to low pressure. And it just depends what part of the plant body is considered sink tissue. Like, in this case, it was a potato tuber. Maybe it's a developing flower or some kind of fruit that's developing, an apple, for example. All right, so as long as you can see that there's this pressure gradient from source to sink, you will have this movement. It's passive. There's no ATP in the sieve tube element. It's just a passive flow based on a pressure gradient. So it's actually quite ingenious. Just make, create a pressure gradient, and there you go. You don't have to mess with any ATP expenditure or any other more complicated system. So going back to the full picture, just let me point out, again, in the sieve tube element, there are no membranes being crossed. Because if you look at the overall water potential up in the source is negative 1.1. If you look at the overall water potential in the sink, it's negative 0.4. So if you just look at that and aren't thinking too critically, you might go, wait a minute, negative 0.4 is less negative than negative 1.1. We should have water going, in this case, up the image, not down. But remember, we talk about water potential, how will water move? We're thinking about through a selectively permeable membrane, part of the process. And there's no membrane, okay? So I've had students get confused on that before. There's no membranes being crossed, just this open system. So therefore, you don't have to think about the laws of osmosis in the sieve tube. We do everywhere else, but not right there, because there's no membranes being crossed as we go from sieve tube element, all the way to the source. So just don't get tripped up with that. It's a little confusing unless you start really keying in on the fact there's no membrane, so we're not talking about osmosis in the sieve tube element. Okay, so xylem and phloem work on pressure gradients. It's just kind of the opposite, where in the xylem it's actually a tension gradient originating in the leaves. With the phloem right next door, it's a pressure gradient, positive pressure in the source, lower pressure, still positive in the sink. Right? So it's kind of odd that they're right next to each other, and yet they work with a tension versus a positive pressure gradient between when you compare the two. Questions on phloem translocation, the process of it anyways. Questions on that? Okay. So this is a good slide to study off of and work through the process. Okay, well, there are two definitions I'd like you to also be aware of. Allocation is one. So I've seen students and even professional writing kind of confuse the two. So we're going to use the definitions that are in these PowerPoints. And so the word allocate can be thrown around for other purposes than what I'm going to be talking about here. And I think that's part of the problem why it gets a little confusing. But when you look at the source and the sink tissue, you have this idea that, okay, what is the plant going to do, whether it be source or sink, with that sugar that's getting made? What are, some of the options? And so it could be, especially, toward the end of the growing season, there could be like, well, I'm going to store it away. I'm going to turn it into starch for long-term storage. So that's an option. And it can be both the source and the sink tissue doing that. Utilization, that's fair. The plant has to stay alive, so they need sugar to run through glycolysis and other physiological pathways to get their energy needs met. So, we do that too. After we eat a meal, we're busy doing glycolysis and all that to get our ATP. Or if you're a source leaf or source tissue off in a leaf, you might say, you know what, I'm going to transport it to a sink. So those are some options. So what we're describing here is the metabolic phase. What's going to happen to that sugar? Should I store it, run a pathway that causes starch synthesis? Should I use it right away so that gets shunted over into glycolysis? Maybe they have to repair some damage. Maybe they have to build some part of their cell or tissues. Maybe they're going to do some growth or repair. Okay, but these are metabolic pathways that we're thinking about. So it's the metabolic fate, and it can be any part of the plant body. source tissue, sink tissue. And so this is how we're going to use the idea of allocation. We're allocating, so much sugar goes into starch. We're going to allocate another proportion to go into glycolysis. Another portion would get sent on to the sink tissue if you're a source. Okay, now compare this to partitioning. So often I see that these two words, partitioning and allocation, used in a very confusing way out in the general, just ordinary writing, non-science, well, even within science writing. Partitioning technically is the differential distribution of that sugar within the whole plant body. So basically, partitioning is looking at the source going, okay, where do I send this sugar? The sugar that I have available to send off, who gets it? Should it be the root? Should it be the baby flower, the fruit? Who gets it and how much? Will get, 10% to roots? Or should I send 50% to the flowers? And so this, the distribution of the sugar is the idea of partition, right? And what they have found is that sink tissue is competitive in the sense that they often refer to sink tissue as, oh, they'll say that sink tissue, boy, that's a really strong sink. And they'll say, oh, this one has become a very weak sink. And so they'll actually use those terms. And what they're getting at here is that some things really seem to be able to draw, like if it's a strong sink, wow, we're getting a lot of sugar, whereas these other sinks aren't getting much. What's going on here? And so they realize competitive. Part of the reason they're competitive is sometimes it's relative, like how close physically is the sink to the source, because then it's a very short pathway, and so that sink tissue would tend to get a lot of the sugar because it's relatively easy just to send it a short distance. They've also noted that the vascular connections, like how are the veins and the vascular connection between the source and the sink? Is it really straightforward? Is it more branched, complex? That can play into it. And then they've noticed that strong sinks really can kind of influence and affect the pressure gradient, and that can influence how much sugar they get. I'll explain a little bit more about that. And so they've come up with this little equation. They say that the sink strength is equal to the size of the sink and the activity of the sink. When they say activity, they usually mean metabolic activity. Like how is it going to, if it's really super active physiologically, it's not going into dormancy or anything like that, then they don't have good, strong sink activity. So if you can imagine the idea of like those baby tubers, let's say it's, toward the end of the fall, that potato plant is starting to grow. And let's say some of the tubers are pretty decent size, some are baby tubers and not quite there yet. Well, they will have different sink strength. And then a mature tuber will have different sink strength. So a mature tuber might be going, you know what? I'm pretty much done. And so they don't demand, so to speak, the sugar. And so their sink strength gets decreased. Whereas a medium-sized tuber that's like ramping up, I have to double my size by the end of the fall or the growing season. So they have a decent sink size physically, plus they're super active. Whereas a baby tuber, they're probably going to be super active, but they're not very big yet. And so you can start to see where there's different so-called sink strength, depending on how active they are, how big they are. And then they can actually kind of communicate back to the source, often through affecting the pressure gradient. I'll explain a little bit more about that. This image that I put here is the idea of how people have taken advantage of this concept. And if you know what you're doing, you can kind of manipulate partitioning by pruning your plant. And so here I took this off a website on how to prune apple trees. And there's a whole science and art to that whole process, especially if you are confronted with an old apple orchard, then you've really got your work cut out for you. But basically, what you're doing is you're impacting partitioning very strongly, especially if you prune as dramatically as what's shown in the picture. And so, if you prune an apple tree, sure, you're going to have less apples on that tree, but each remaining apple that got left on the tree, it's going to be bigger than if you hadn't pruned. It's going to be bigger, it's going to be sweeter. So they get more of the lion's share of the sugar. They get, the partitioning, you've influenced that to those remaining apples. Now, if you had not pruned, you'd have a lot more apples. But if you're trying to make a profit out of that, probably not work as well because they're going to be small. They won't be very sweet, and you won't get a very good price for that. And so humans like big, sweet apples for the most part. I mean, there are some like Granny Smiths you want a little bit tart for certain types of pieces, but for the most part, most apples sold are for the sweetness, not for baking apples. So this idea of pruning and the apple orchard or the apple tree is a really good example, but you can prune any plant, really. It doesn't matter. Any pruning that you do, you're influencing partitioning for sure. And so there's been a lot of interest in this. They have been very, very interested in looking at how does a plant decide, so to speak, where to send the sugar, and can we influence that? So there's been a ton of research into the biochemistry of all this. They're making some progress, but when I was looking more carefully at some examples to provide, I was kind of surprised that they hadn't made a little bit more progress than they have. So, but they're still really, really working on it because this idea of, can we influence where the sugar goes? So if you are trying to make a profit off of, growing pea plants or bean plants and you want lots of fruit production, well, then you want to try to get the sugar into the fruit production more to a point. I mean, you don't want the roots to starve and not grow as well. I mean, there's this trade-off, right? And so they know that. Like, you can't just send everything into the possible part of the plant, obviously. You still need a good root system, you still need enough leaf tissue. But they are trying to find ways to manipulate that so that you can ramp up the production of what you want from that plant, whether it be peas in a pod or apples or whatever. So what they're showing here, I thought this was a nice visual because it clearly shows difference between allocation versus partitioning in the true like plant physiology sense of the definitions. And so on the left, you can see allocation is looking at, they're trying to show like physiological pathways. What should we do with that sugar? And then partitioning is much more like, let's zoom out, look at the whole plant body. Now, where should all that sugar get sent? Where should it get partitioned to? Should it partition into the roots? Should it partition into fruit, flower, baby leaves? Okay, so this research is hot right now. They're really interested in either through I actually saw quite a few papers on genetic engineering that so they're looking at trying to create pathways that kind of force more partitioning into certain plant parts. And so what they are finding through all of this research is that the sick tissue really can communicate back to the source on its status. And it can do it through a variety of ways. The seems like the most common strategy is to do these pressure gradient changes. So what I mean here is, let's take the example of, let's say that medium-sized potato tuber is like, man, I need to double my size in just a few weeks. I'm just gonna grab all that sucrose that's coming down from my source tissue and pack it away as quick as I can. So if you rapidly unload like that and pack it into the potato tuber, what you're doing then is you're lowering pressure pretty dramatically in the sink, right? The subtube element sink tissue. And so you created a steeper gradient if you all of a sudden suck all that stuff, but bring out that sugar that lowers your size of tea, and then you can pressure grade between the source. And that's actually a way to communicate. You know, if the sink tissue all of a sudden there's a drop in pressure, the source is like, oh my gosh, you need a lot more sugar right now. Okay, and then it'll send it, and so you'll see when they track it, they'll show like in this case the potato tuber really taking that sugar and packing it away, and then track the source tissue. They can see the source tissue ramps up loading of the sucrose into the vein. Like, all right, you know, speed up. We got to get this going. And so you can actually see that through careful study. It's really quite amazing that they can track that and see that. And so the source will just, yeah, okay, I'll get you more sugar. Here you go. And then eventually that tuber is going to get to its mature size and go, okay, I'm doing pretty good now. I'm gonna not, you know, demand as much. And then that source tissue will perhaps divert now into sending its sugar to a different sink that means it has a stronger sink strength. So that pressure gradient is a pretty straightforward way of communicating that to the source. Then they've looked at, is there anything else flowing maybe in the phloem tissue that can come from the sink area and reach the source tissue? And so they've been hunting for that. And so what they're looking at, of course, the sucrose concentration, that kind of goes with the pressure gradient change. Because if the sink tissue all of a sudden really draws heavily on getting the sucrose packed away, that's going to be a signal. But they've looked for hormones. Could there be any hormones being sent from the sink to the source? And there's been some limited data showing that hormones could be involved under some conditions and with some plants. They've looked at small pieces of RNA, messenger RNA. They've looked at proteins. They've looked at ions as well. And so they're trying to tease this apart. They don't have solid answers for all of this yet, other than the pressure gradient. That seems pretty established. But some of this other stuff, they're still trying to hunt down the way it actually works. But these are some of the topics there's hints that there is some other stuff going on, which kind of makes sense. When you think about it, a plant should have more than just a pressure gradient change to kind of assess its situation. Because this is pretty dire. You want, of course, seeds to be made and produced. If that goes wrong, you can't pass your genes on to the next generation and you go extinct. So it makes sense that, I understand why they're hunting for some other ways of communication, because this is such an important thing for plants. They have to get this right or they could end up going extinct. And so it does make sense that there's more than just a pressure gradient change. So the story is still unfolding on that. One paper that I did read, it was a review paper. I'm not going to ask you to read it. This is just excerpts from it, but kind of a nice summary. This was in the introduction section of the paper, and I'm just talking a little bit about the idea of what that sucrose is going to be used for, metabolically, as well as partitioning idea. And so if you could read through this in your own time as you're studying, it pretty much explains or touches on everything we've touched on, just, paragraph format. Of course, if you think about like wheat plant is, billion-dollar agricultural crop worldwide. And they're, again, getting at this idea of, obviously we want wheat grains that are packed with nutrition, including sugars. And so figuring out how to get this to happen and can we manipulate it. And then the concluding remarks at the end of the paper, they're saying that, there are some genetic modifications that have been done that have some limited impact, but the authors are basically saying that, we need to keep looking at this and doing research to optimize our ability to manipulate the plant, or especially if you're trying to sell it for profit. And feed how many billions of people by the year 2050? What's the number now? What are we at now? 8 billion? Are we at 8 billion already? Are they saying 10 billion by 2050 or something like that? If you go to a plant biology conference and you go to the sessions where they talk about this, like, and right now they're using the benchmark of the year 2050, you walk out of there going, oh my gosh, I need a beer or something. It's like stressful. Like, how are we gonna feed all these people? And so, they've come to the conclusion that we need many strategies in order to do this. And so, partitioning is one idea, better understanding. Okay, questions on Phloem translocation, allocation, and partitioning? Like they have a oh, very good question. So then, if you have that potato tuber getting mature and it's like, okay, I'm done growing, I've packed away all my nutrients. So what happens is essentially what you see happening at that particular sink is you don't have this unloading, and that stops. So this stops. And so then the sucrose stays there, doesn't get unloaded. We call it unloading into the tuber. It just stays there. And then that causes, there won't be a pressure drop anymore. In fact, it could actually build up there, where now you could actually get a situation where if there's no unloading, your size of P value could actually start to increase after things, right? And then what if the size of P starts to equal out the size of P in the source? Then we say the pressure gradient dissipates. And then in that case, it would tell the source, you know what? I'm done. I'm done. I'm no good. I'm not too good. And so just that gradient disappearing or dissipating is the signal back to the source. Oh, okay. I'll send it somewhere else. Okay. Other questions? Okay, so transpiration, Phloem translocation, really, really important concepts to think about when you're growing plants, for sure. The other huge concept that we need to talk about is photosynthesis and how the plant is creating all this sugar. And so the next slide set should be posted looking at the light reactions of photosynthesis. So that's the next slide set I want to get into right now. So with the light reactions, we tend to break down photosynthesis in like two different aspects. The light reactions are where the light energy is actually being used. And then the second part of photosynthesis, they often refer to as like, they call it carbon fixation, or the biochemical reactions, which, I mean, we're all biochemical reactions. But anyways, so we're going to look at the light reactions. And this quote is very appropriate in terms of, with the light reactions, we're going to be learning how electrons are getting passed from molecule to molecule to molecule. And here they're saying, life is driven by nothing else but electrons, by the energy given off by these electrons, while cascading down to the high level to which they have been boosted up by photons, which is light energy. An electron going around is a little current. What drives life is thus a little electric current kept up by the sunshine. All the complexities of intermediary metabolism are but lace work around the sky. Very poetic, wasn't it? So, and very accurate. So what we're thinking about here is this light energy actually causing electrons to be lost from one molecule or donated off from one molecule and getting sent over and accepted by another. And so a refresher, I'm assuming you've had a little bit of information about light in your process so far here and or in K through 12, but we're looking at this idea that light is electromagnetic spectrum. It is odd in that it can behave as a wave under some conditions and as discrete particles under others. I'll explain. For humans, the visible spectrum of light is in the range of 400 to 700 nanometers. So wavelengths are expressed in the nanometer idea. I'll talk to you a little bit more about that. And then this equation at the bottom is just to get you thinking about the idea of how frequency and wavelength are related to each other. They're inversely related to each other. So V, the fancy V is frequency, is equal to C, which is the speed of light, divided by lambda, which is your wavelength. Okay, so V is frequency, C is the speed of light, and lambda, so V is equal to C divided by lambda. And so that's why we say they're inversely, because the lambda is in the denominator. So the wavelength is in the denominator, and that makes it inversely related to the frequency. So I am not a physicist, so that I'm not going to get into more details than that with regard to light energy, other than a few points of clarification. Here, you're just looking at a very human-centric way of, we can see from 400 to 700. Other organisms are capable of seeing on either end of that more than what we can. So this is a very human-centric diagram. But if you shine light through a glass prism, it will break into these different qualities or colors of light. They correspond to different wavelengths. And so red, orange, yellow, green, blue, indigo, violet is the order from largest wavelength to smallest. And so, looking at a rainbow after rain, that's what you're seeing. And so you can see there are other wavelengths coming from the sun. There's the X-rays, there's the ultraviolet, we just don't pick up on that with our ability, our body, and our eyesight. Okay, and then the top two are just trying to show you how wavelength and frequency are inversely proportional to each other. Okay, so what we kind of zoom in on for the light reactions. When the light energy is striking the plant body and striking the chlorophyll inside the chloroplast, then we start thinking about a stream of particles. So when sunlight energy is just moving through the atmosphere, it behaves as a wave of energy. But when it hits a solid object, then it starts to behave as a stream of particles. And then we start referring to that energy as photons. The energy carried by a photon we refer to as quantum energy. Okay? And the quantum energy can be defined as hc divided by lambda. And remember, c divided by lambda is the definition of frequency. So you can express it either way. You can say quantum energy is equal to hc divided by lambda, or quantum energy is equal to h times frequency. H is Planck's constant, if you're curious. Okay. And no, we're not going to do any math to figure out quantum energy, but I'm trying to remind the part of your brain that might have learned this already a little bit of this idea of photons and quantum energy striking the plant body. Okay, so that light energy is striking the plant. Specifically, we're zooming in to the chlorophyll. Yes. Not math per se, but describe it in words. That's what I'm more interested in. I'm not going to test your math ability because that's irrelevant to me. What's relevant is you are creative. You understand. Yeah, like if I give you a scenario, if sucrose is getting your Well, it gets more negative, but I wouldn't give you equations to solve. I wouldn't do that. So, yeah, and as you're studying any of this, if you have questions like that, just, and I'll let you know what level or how much I want you to know. So keep asking questions like that because I'm happy to direct you so you don't get off into the weeds, so to speak, on what to study and whatnot. All right, so we're zooming into chlorophyll, that wonderful, amazing pigment that is able to absorb certain quality of sunlight energy. And so what we're going to find is that the energy coming from that photon from the sun is actually going to get transferred and excite an electron. And that electron will get boosted to a higher energy level. It'll get excited, okay? And in that state, that's when we can start to see an actual chemical equation. If everything goes okay, we can end up with a chemical equation where that electron will get given off to another molecule, okay? So we're thinking about this idea of energy, that light energy striking the chlorophyll, and it's often shown something like this, where if the sun is striking, the sun energy is striking the chlorophyll, what you're seeing here, if you take chlorophyll, chlorophyll suspension, and you put it into a spectrophotometer and you run an absorption spectrum, that's what you're seeing in the figure B portion. Okay, so figure B is your absorption spectrum. Okay, so you can see this double peaks. You have these peaks in two different spots. The red quality of light and the blue quality of light. Okay, so those wavelengths of light, you can see the wavelength scale on the far right there. So then if you match that up to the figure A, that's your kind of your energy. They're representing what happens to those electrons when they get hit with red light or blue light. And you can see there's actually kind of a big difference. If you take blue light and strike the plant with blue light, you can see that the electrons get really excited, okay? If you hit the plant with just red light, they get excited, but to a lower level, all right? So we know that plants preferentially absorb red and blue light according to the absorption spectrum. You can see this big absorption peak in the blue quality of light, big absorption peak in the red. So anytime you run these kinds of absorption spectrum, you see these peaks, that's telling you, oh, that's the quality of light that organism preferentially, some pigment is capable of absorbing. Okay, in this case, we're looking at chlorophyll. And chlorophyll A is really well designed to maximally absorb red and blue light. But it actually has slightly different energy excitation properties. The blue light actually gets it more highly excited than the red light. So they've been looking at that with regard to what qualities of light. That's why you often see grow light. They'll say they're enhanced in the blue and red region. That's why. It's because they know that that causes the chlorophyll to kick into the light reactions. So we're zooming into chlorophyll A, and mostly chlorophyll A and chlorophyll B is what we'll think about for terrestrial plants. They will be using those pigments primarily in the light reactions. So on this figure, the chlorophyll A is being shown on the far left in figure A. And so you can see that there is two kind of parts to the pigment. So you have the so-called porphyrin head region, which is this more complicated region here. And you can see that there is a magnesium bound or chelated in the center of that porphyrin head. And then the phytol tail is this long, skinny thing. So that's your phytol tail. So the phytol tail is actually embedded in the thylakoid membrane. Okay, the thylakoid membrane inside the chloroplast. So this phytol tail is stuck all the way through the membrane, and then that porphyrin head region kind of sticks off the membrane. okay? So chlorophyll A is written when you have a methyl group right in that position, that's chlorophyll A. If you replace that methyl group with this aldehyde group, that's chlorophyll B. That's the only difference. That's it. These other pigments that you're looking at, bacterial chlorophyll A, that's found in certain bacteria that can do photosynthesis. Carotenoids are being shown in figure B. Talk about them. Bilin pigments are often in algae, variety of algae. Okay. But we're going to zoom into chlorophyll A and B. But be aware, chlorophyll A and B, all the so-called higher plants, so the terrestrial plants, gymnosperms and angiosperms, a bunch of different algae. CD is cyanobacteria, so they have chlorophyll A. Chlorophyll B, again, pretty much all the plants you can think of, and a bunch of different algae. But then there's also chlorophyll C and chlorophyll D, just to be aware that there are, some other versions besides C. Chlorophyll C you find in the diatoms that we looked at earlier. We didn't really talk too much about dinoflagellates, but they're also having chlorophyll C, brown algae, and then the chlorophyll D, red algae, and cyanobacteria. So there are other versions of chlorophyll. And then just be aware, so far anyway, the carotenoid type pigments, those are the orangey yellow pigments that you see, like a carrot taproot is chock full of carotenoids. Pretty much any photosynthetic organism has carotenoids. I have yet to see anyone declare that, oh, this photosynthetic organism doesn't have carotenoids. It seems like it's just universal. Every single plant or organism, including the bacteria that can do photosynthesis, they have carotenoids. And so carotenoids provide kind of multiple roles. They're very important. They can help with light energy absorption, but they also protect the plant. And I'll talk more about that. Okay. So what we're going to get into is looking at leaf design next, and then we'll get into, okay, what is really happening when we zoom into the chloroplast and look at the light production. We'll start that on Wednesday.